Assalamu alaikum. So this lecture will give us an introduction into the human genome, okay, and different components of the human genome. Notice that the chapters or the pages in Cooper's textbook are scattered all over the place. So what I've done is that I've tried to organize the lecture to highlight the most important topics and information that you need to know. So the main resource, of course, as always, is this lecture itself. Let me start by talking about the Human Genome Project. Now, this project was launched in 1990. There was a lot of talk about it, like 10 years before 1990, in 1980s. It was a huge project. It would cost about, it was estimated that it would cost about $3 billion, that it would last 13 years, and it lasted about 14 years or 15 years. And it was a multinational project. It started in the US, of course, and then other countries, like 7-8 countries, joined in that mission to sequence the human genome. Meaning that the whole purpose was actually to know the sequence of letters in the human genome. Major outcomes of the Human Genome Project, when it was done in 2004, is to identify the human genes. And it was estimated in 1990 or 1989. That was the year that I started studying actually my bachelor's. It was estimated that the number of human genes was 100,000 based on complexity in comparison to other organisms like viruses and bacteria and yeast and so on. So it was thought to be about that number of genes was 100,000. And then after going through the Human Genome Project, like every few years, they would say it was an overestimation. The number of genes in the human genome is like 50,000, and then it went down to 40,000, and they were saying that with astonishment. And then it went down to 30,000, and then again 25,000, and finally, at the end of the Human Genome Project, they knew that it was about 22,000 genes. Now, and they also realized that the coding region, that is the region that codes proteins, represents about 2% of the human genome only. And they thought that the other 98% was quote-unquote junk DNA. And they knew that this 2% represented mainly, or it encoded transcription factors. Okay, they also realized that there were a lot of regions called enhancers. We'll talk about enhancers in the transcription lecture. They knew that there was a lot of control regions like enhancers within the genome. Other things that they were able to accomplish by the end of the Human Genome Project is that they completed sequencing the other genomes of other organisms like mitochondria, different types of bacteria, yeast, Drosophila, that is fruit fly, yeast, I said yeast before, C. elegans, that is, which is a worm, mice, and chimps, and etc. So they were able to do that, and they were able to do that because they developed a lot of technologies. And remember that any advancement in science or medicine is always associated with technologies, and technologies really are what make us do wonders in science and medicine and other fields, scientific fields. So they also were able to open discussion of legal and ethical issues associated with the human genome. That is it ethical to sequence a human gene without telling him? Is it ethical to tell other people what mutations different people would have, or how about telling a family that they have a certain mutation, for example, okay, and so on. So there were a lot of issues. Also, there were issues related to patenting the human genome. How would you imagine patenting a DNA sequence so that no one else would benefit from that DNA sequence commercially? It was quite amazing. Anyhow, so it was a huge project, and there was a lot of competition between different centers. Long story. Anyhow, so that was the Human Genome Project. Now, they were able to tell that the human genome was composed of about 3.2 billion base pairs. In fact, it's about 2.9. We know now that it's about 2.9. That we have about 20,800 genes so far. That's what we know so far. And these genes and these bases are distributed throughout 46 chromosomes. Now notice in mice or mouse genome is basically very close to the human genome. Number of genes is the same, almost the same, about 20,000 some genes. Number of chromosomes is very close. But you look at fruit fly, for example, Drosophila. There are 13,000 genes. I mean, you look at the worm. This is a very famous worm that we use in laboratories, C. elegans. 19,000 genes. I mean, really, what's the difference between worms and humans? We're talking about 1,000 genes only, which is quite amazing. So you can see the comparison of genomes. Of course, this is not for you to memorize. Just you need to know the number of genes in the human genome, of course, the base pairs in the human genome, and so on, but not other organisms. Now, this is comparison of nucleotides, number of base pairs in different species, archaea, bacteria, plants, for example. You can see the genome, the sizes of genomes are widespread. So you have small genomes and you have large genomes. Same thing with fishes, mammals. Human genome lies almost in the middle. Okay, so you can see, but what's noticeable is that the more complex the organism becomes, the more complex or the larger the genome is. But that's not necessarily true. I mean, if you look at protozoans, for example, and amoeba has larger genome than humans. Anyhow, so this is just for you to to know, not to memorize, of course. So this flow chart shows us the different components of the human genome. So again, the human genome is composed of about 3 billion nucleotides or base pairs, and there are about 20,000 genes or so. And if you look at these genes, there are about 40% of the human genome is basically genes and gene-related sequences, whereas 60% of the human genome contains intergenic DNA, that is DNA within genes. But if you look at human genes, or the 40%, about 2% of the human genome is composed of the protein coding regions, that is exons, that is the regions that make mRNA, messenger RNA, that makes proteins. Okay, and 38% is non-coding RNA genes. Now, if you look at these non-coding regions, they are really mainly introns. Okay, about 35% of the human genome is introns, but there are other gene-related sequences like known as pseudogenes. Pseudogenes is fake genes. Gene fragments, introns, of course, untranslated regions, and we'll talk about untranslated regions later on. But if you look at the 60% of the human genome, it's mainly you have 5% unique or low copy number, but the majority really is basically repeats. Okay, that is regions that are repeated. So it's the same sequence in the human DNA, but it's repeated in different places or in the same place. So basically, if you look at this 55%, it can be divided into two types. You have tandem repeats, and what tandem repeats mean basically is that it's the same repeat, but they are very close to each other, or it's one after the other. So they are consequent to each other. They come one after the other. Okay, so about 10% of the human genome is basically tandem repeats, and these tandem repeats can be divided into satellites, mini satellites, and you have micro satellites or STRs, or short tandem repeats, and we'll talk about these three. Now, the majority of the human DNA basically is composed of interspersed repeats. So this is what this means. It means that it's the same sequence, but it exists in different regions of the human genome. So they are far away from each other. They are separated by large distances from each other. So in the human genome, you can find a repeat, for example, on chromosome 1, and you can find the same sequence on chromosome 5, and you can find a third one on chromosome 12, and so on. So these are dispersed or interspersed repeats. Now these repeats are divided into two types as well. The majority is what is known as retrotransposons, and we'll talk about these at the end of this lecture. So whenever you hear the word retro, you know that it means that the source of the sequence is viruses. Retroviruses, that is viruses with their genetic material being RNA. And about 3% of the human genome is basically DNA transposons. Now, if you look at these retrotransposons, you have large portion of it being long sequences, and about 13% is short sequences, and you have 8% being retrovirus-like elements. And we'll talk about all these as well. Now, what's important as well is that 5% of the human genome contains sequences of non-coding DNA. Okay, meaning that, again, DNA that does not code for anything. And these are highly conserved. Okay, and that gives us an indication that these sequences are important, and that's why they have been kept throughout these millions of years within the human genome. Okay. Otherwise they could have been lost. Now, please pay attention that all these numbers are approximate. Okay, so I'm not going to ask you, or I'm not going to say, do retrotransposons constitute 42% of the human genome, 43%, or 40%? You know, I'm not going to put in close numbers. Okay, so numbers will be far away. So, but you need to know that, for example, about 40% of the human genome is made of retrotransposons. So let's talk about, you know, all of these sequences. But first, let me introduce another project. So this project started at 2003, huge project, again, multinational, a lot of money have been put into this project. So by getting close to the end of the Human Genome Project, the US government started another project to understand the different components of the human genome, you know, with the advancement of technology. And this project was called the ENCODE project. And basically what they wanted to see is, or what they wanted to do is to analyze the different DNA elements. And again, whenever you hear the word elements, you know that we're talking about a sequence of DNA, or a certain region in DNA or RNA. Now, a major outcome of the ENCODE project so far is that they knew that 80% of the entire human genome is relevant. Okay, so they thought that only 2% of the human genome is important, and the other 98% is junk DNA. Okay, but then they realized that actually 80% of the entire human genome is relevant. About 75% of the human genome can be transcribed. Okay, they could not imagine that. They thought that only 2% of the human genome was transcribed, but then they realized that 75% of the human genome was or is transcribed, which is amazing. Now, in addition, you have other regions that are important. They are binding sites for regulatory proteins, or they are associated with other biochemical activities. So if you look at the table here, and this is taken from Cooper's textbook, so they realized from the ENCODE project that there are about 20,000 genes plus 20,000 plus genes. You have something known as short non-coding RNAs, and actually numbers are larger than that. And you have long non-coding RNAs as well. Okay, and you have pseudogenes. Pseudogenes means they look like genes, but they're not genes. So what are pseudogenes? Pseudogenes are fake genes. So they look like genes. They can be homologous to other genes, to existing genes. Homologous means like similar sequence, DNA sequence, like the globin genes that are responsible for making hemoglobin and myoglobin and so on. So they are duplicated, but they are not expressed. They do not encode proteins. Okay, there are a lot of them in our genome as well. Now, the percentage of genome that is transcribed into RNA is about 75%, and the percentage of genome binding transcription factors, okay, we'll talk about these transcription factors, it's about 8%, and this is a huge percentage actually if you think about it. Now, this is a new pie chart showing the different components of the human genome. So introns make up about 1/4 of the human genome. The protein coding genes, that's about 1.5% only. You have lines and signs, okay, and we have other types of retrotransposons, the retroviral-like transposons or regions. We have DNA transposons, they make up about 2.9%, 3%, and so on. So we have other repeats and what have you. So we will talk about these throughout this lecture, okay? So it's not going to be something like foreign language to you, okay? So let's start with tandem repeats. That is, repeats that you have them one after the other, okay? So they are sort of like sequential. Okay, now, so you have something known as satellite DNA, or they're known as macro-satellite DNA. Okay, and these are basically regions that are composed of about 5 to 300 base pairs, and these are repeated like millions of times. Okay, and they make up about 10% of the human genome. And there are different types of satellite DNA or macro-satellite DNA. Probably the most common are centromeric repeats. Centromeres are these regions that exist within chromosomes. They divide chromosomes into two parts, the short arm and the long arm. Okay, the P region, petite, and the Q region for grande. Okay, and these centromeric repeats are rich in A's and T's, and they are important for the separation of chromosomes during the cell cycle in mitosis or meiosis. So they are the regions where they attach to the long filaments and you have the chromosomes pulled to either pole of the cell. Now, these regions, by the way, can be stained through a technique known as fluorescence in situ hybridization. So you know what hybridization means. In situ means in place. So what you're doing is you are staining a certain thing in its place using fluorescence methods. So basically the idea here is that we use a DNA probe, and this DNA probe is fluorescently labeled, and then it targets chromosomes, and you have this hybridization between the probe and the chromosome, regardless of the characteristics of these chromosomes. So right here you can see a chromosome, and the centromeres are fluorescently labeled. Okay, so this is a very common technique that is used in medical laboratories and in hospitals to stain chromosomes or different regions of chromosomes. Now, there are other repeats, and we talked about these repeats before. These are the repeats that exist in telomeres. Now, telomeres are, again, they exist at the ends of chromosomes. Telo means end. Okay, so these are the regions that are located at the tips of chromosomes. This is another image of fluorescence in situ hybridization, where you have telomeres being stained. Now, of course, telomeres, as you know, they're composed of a repeat, that is TTA-GGT, and or TTA-GGG without the T at the end. It doesn't matter because it's a repeat. Anyhow, and these are repeated like hundreds to thousands of times at the termini of chromosomes. And there is a 3' overhang. Okay, so right here at the end of the chromosome, that's where you have the lagging region being extended, right? So you have this extension right here that is created by the telomerase. Okay, now remember how I gave you the example of telomeres after being elongated, and in order to make sure that chromosomes are stable, it's like sewing a button in a shirt or something. So at the end you have this extra thread, and you just tie it, okay, so that the button would stay in its place. So this is the same thing. You can visualize it as the same thing. So at the end of the chromosome, it's tied up, and this is done also with the help of a protein known as shelterin. So what it does is like it shelters chromosomes. Okay, so it protects the termini of chromosomes from degradation. Now, there's something fascinating that I found, and it's not in your Cooper's textbook, and that is telomeres can be transcribed. Now, in the past 15 years or so, I've been teaching molecular biology, and I always said that telomeres and centromeres are not transcribed. They are heterochromatins. They do not contain any genes, but actually now we know, because of the ENCODE project and other work, and development of technologies, that telomeres can be transcribed as well as centromeres, but I didn't talk about centromeres being transcribed in the previous slide, but telomeres can be transcribed. Okay, it's amazing. So you have the formation of a telomeric repeat containing RNA, also known as TERA, and this RNA it belongs to the class of long non-coding RNA molecules. And these RNA molecules that are transcribed from telomeres, they have important functions in maintaining the integrity of chromosome termini. So they work similar to sheltering. They can also regulate telomerase activity. They maintain the heterochromatic state of telomeres. So remember, we have euchromatin and we have heterochromatin. Heterochromatin is where you have DNA highly packed. Telomeres are maintained in the heterochromatic state, very highly packed, and this RNA tera also can protect DNA from deterioration and degradation or fusion with neighboring chromosomes. Now, there are genetic abnormalities whereby you can have chromosomes fused with each other, and it would be fascinating to know what the role of tera is in chromosomal fusions. So this is really fascinating and up-to-date information. Now, mini and microsatellite DNA sequences, they are highly repeated, okay, but they differ in size. So, for example, mini satellites are also known as variable number tandem repeats, VNTRs. So you have a sequence that is made of, let's say, about 20 base pair sequences, 20 to 100 base pair, and this sequence is repeated several times, and they are tandemly repeated, so they are close to each other, one after the other. Microsatellites are repeats that consist of about 2 to 10 base pairs, and they can also be repeated several times, as you can see in this example right here. So mini satellites, or VNTRs, they are each repeat contains about 20 to 100 base pair sequence, and it can be repeated 20 to 50 times. On the other hand, microsatellites, they are repeated 10 to 100 times, and they have this size, 2 to 10 base pairs. Okay, so you can have, for example, since we are diploid, or our cells are diploid, you can have on a chromosome this repeat right here, the CA repeat, so CA, 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 and it's repeated 16 times. On the other chromosome, you can have the same exact repeat, but it's repeated 14 times instead of 16 times. So you can have a lot of variation, even individual. Now, this is a cause of polymorphism, that is, different shapes of DNA sequences. So you can have, for example, different sequences of VNTRs and STRs on the on homologous chromosomes, like I showed you in the previous slide. And this has been helpful actually in forensic testing, because we can differentiate samples of individuals from each other based on this polymorphism. So here's an example right here, a few examples. So you have individual A, you look at STRs or a certain VNTRs. On one chromosome, you have 3 repeats. On the other chromosome, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 repeats. Okay. Now, on the other hand, you have individual B on homologous chromosomes. The same exact sequence exists on the same location, on the same chromosome exactly, except that the number of repeats is different. So on both chromosomes, you have 6 repeats rather than 3 or 10. So this is individual A. You have this repeat is the one that is repeated 10 times, and this is the one that is repeated 3 times. Now, in this individual, there's only one band. If we do RFLP, or if we do PCR, and we'll talk about PCR later on, and there's only one band, which is the 6-time repeated VNTR. So look at these different individuals looking at a certain VNTR or STR. You can see how each individual has his or her own molecular pattern, and this is what is known as molecular profiling or fingerprinting. Now, we can use VNTRs and STRs in paternity testing, in forensic medicine, in diagnostic medicine as well, and this is an example. Okay. So right here in this family, okay, this is known as a pedigree. So what you have in here, you have square representing a male, a circle representing a female. So you have male and female, they're married, they are connected to each other, and they have this little daughter. Now, and you have another family, female, male, so they get married and they have this boy, and this boy gets married to this girl right here, and they have lots of kids, or أولاد, in other words. So they have this female, male, 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 female, male, and female. Okay. So if we look at this family, and if we look at a certain VNTR, for example, we would notice that there are 4 different alleles. So alleles is basically type of a gene or type of a DNA sequence. So they have 4 alleles. Allele number 1 is when they have 6 repeats. Allele number 2 is having 4 repeats. Allele number 3 having 3 repeats, and allele 4 is having 1 repeat only, or one of such sequence. So by doing RFLP or by doing PCR, we get the following. So these patterns represent the person that is located on top of them, okay? So here we have the grandfather, okay, of the first family, and this grandfather has allele number 1 and allele number 2. So he has these 2. Now, this grandmother has allele number 2 and allele number 3. So you would expect this daughter to have one of these 2 from her father and one of these 2 from her mother. And that's true. In fact, she has allele number 1 from her father and allele number 3 from her mother. Look at this family now. Let's look at this grandmother right here. She has alleles 2 and 3. Look at this grandfather. He has alleles 1 and 4. So you would expect this boy to have either one of these 2 alleles and either one of these 2 alleles. And that's true. He got allele number 2 from his mother, and he got allele number 4 from his father. Okay, now let's look at these individuals right here, these children right here. So you would expect them to have either alleles 1 and 3 from the mother, or alleles 2 and 4 from the father. And you can see the pattern in here. Okay, so this is from the mother, this is from the father, and so on. Now, so how can we differentiate these 3 individuals? How can we differentiate these 2 individuals in this case? Well, what we do is we look at another allele, or we look at another VNTR. So or we look at another STR, and so on. So in forensic medicine and paternity testing, they do not rely on one single VNTR or one single STR. Rather, they look at multiple VNTRs and STRs. Okay. Now, so we can use this for paternity testing. Same idea as like I showed you before in the previous lecture, or in this one right here. You can see the pattern between the parents versus children. Okay. So now, one major feature of the human genome is variation. And variation is not only in VNTRs and STRs, rather variation can also exist in single nucleotides. And this is known as single nucleotide polymorphism. So this is a major source of polymorphism. So basically, the idea is that at a single location on a chromosome or DNA, you have the same exact sequence, except that there is a difference in one nucleotide, and this is single nucleotide polymorphism. Okay, so when is a change considered a polymorphism versus a mutation? Well, it is known, this is the thing, if it exists in more than 1% of the general population, if a variation, a genetic variation, a SNP as we call it, exists in more than 1% of the general population, we call it a polymorphism. If it exists in less than 1% of the population, then we consider it a mutation. Okay? So this is an important point that you need to know. Now, so these SNPs are really spread throughout the human genome, and there is one SNP for every 300 base pairs. So we have a total of about 10 million SNPs in our genome. Okay, 10 million SNPs, that's a lot. But only 500,000 are important or relevant. So what do we mean by important or relevant? Well, let's see here. So here we have examples of SNPs. So you have an individual, for example, having this sequence. This person has at a certain location that is known to be variable among individuals in a population. This person has G and G on both paternal and maternal alleles. So we said that this person is homozygous for this particular SNP. Okay, now let's look at another sequence, another location, and we would notice that the paternal allele contains an A, the maternal contains a C. Okay, so we say that this person is heterozygous for this particular SNP. Notice that the distribution of this SNP among individuals in a population is that A exists in 90% of the population versus C, which exists in 10% of the population. So we call this a minor allele. Now, these SNPs in individuals in a population, the G exists in 51% of a population versus T, which exists in 49% of the population. But this individual does not have a T, obviously. But notice that all of the other sequences are exactly the same except at these two positions. Okay, so this is individual 1 looking at a SNP in chromosome 2. Different individuals, 6 individuals. This person has T and C. This individual, same location, same SNP, same position, has C and C. This person has T and T. This person has T and C. So, and this person has C and T, and this person has C and T. Okay, so you can see that these SNPs, mainly they are either T or C. Okay. How are SNPs important? So I said that 500,000 SNPs are relevant. What do we mean by relevant? Well, these SNPs can be divided into two types. They can either be linked or they can be causative. What we mean by causative is that they cause a phenotype. Okay, so they cause a certain phenotype. For example, maybe a phenotype in, I don't know, skin color, maybe a phenotype in something that you can see. Maybe sometimes it's a phenotype that exists when a certain condition happens. So for example, you have two individuals, they look maybe brothers, they look exactly the same, except that when one of them takes a medication for headache, like paracetamol, for example, you see one of them needs just one pill, the other one would need 2 pills. Okay, we wouldn't be able to know unless they go through this certain circumstance or certain condition. So this is a variation. Now, this variation can be relevant, well, because of their need of different doses of medications. So that's what we mean by it causes a phenotype. So that's a causative SNP. Now, it's not a major thing. It's not a mutation that causes a disease. Rather, it causes a certain phenotype. So causative SNPs can be located in non-coding regions, and we call them non-coding SNPs. So they can be located, for example, in the promoter region. They can be located in a regulatory sequence, meaning that a sequence of DNA that regulates, for example, gene expression, protein production. Or SNPs can also be in the coding region. So they exist in the DNA sequence that results in that influences protein production, or the sequence of amino acids in a protein. So we call it a coding SNP. This may change the amino acid sequence. Sometimes this change may not be that important. Okay, so it's not like the protein is defective. The protein, or let's say an enzyme, it might be about 50% less efficient than if it has the other SNP. Okay, and we would not be able to know the person is totally normal. Now, or SNPs can be linked, meaning that they do not cause a phenotype, rather that they are linked to a certain phenotype. So we notice, for example, if we look at individuals having a certain disease, let's say, we would notice that a lot of individuals have with this disease have T's. Okay, it's either C or a T. And these individuals that are affected with a certain disease or condition, they have a T. Okay, whereas in healthy individuals, very few of them have a T. Okay, but most of them have a C. What does that mean? Does that mean that if we change a C, this C to a T, let's say, or if C is mutated to a T, these individuals would be affected? No, not necessarily. Okay, does that mean that if we change T to a C in these individuals that are affected with a disease, does that mean that they would not have the disease? Again, no, not necessarily. True, but what it means is that there is a high frequency that individuals having a disease would have a T. It's just a probability. Okay, so if you know that someone has a T, you would say, you know what, there is a probability that you might develop a certain disease. It does not mean that for sure he would develop that disease. So what does it mean that it's linked? It's linked to a phenotype, yes, or it's linked to a gene. Maybe it's located near a gene that causes that disease. So when the gene is inherited, the SNP is also inherited along with it. Okay, so that's what linked means. And I hope it's clear. Well, how about interspersed repeats? These are the repeats that make up the majority of the human genome. Now, these interspersed segments are also known as transposons, which means jumping genes. Okay, so these are really segments of DNA that can move from one place to another in our genome. And it, by the way, it does not mean that as I'm talking to you right now, I have my transposons jumping or changing places. Same thing with you, by the way. So it's just that these are regions that can change place. Now, the majority of our transposons do not move. Okay, they have lost the ability to move and change places, and they do need help somehow. This is still under extensive research, but in other species, by the way, transposons can jump and can change places, like in pigs, for example, in mice, in plants, and so on, but not in humans. It's not that common. Now, there are two types of transposons. We have what is known as DNA transposons, and they make up about 3% of our genome. Okay, but the majority is basically RNA transposons. or retrotransposons, and they originated from retroviruses, viruses with RNA being the genetic material. So RNA transposons, retrotransposons, they make about 42% of the human genome. The majority is known as long interspersed elements or lines, and they make up about 21% of our genome. Then we have short interspersed elements or signs, and they make up about 13% of our genome. A great example of signs is something known as an ALU sequence, and it's known as ALU because it can be cut by a restriction endonuclease known as ALU. And the size of ALU is about 300 base pairs, and it's the most common sign in our genome. And actually, I was working, when I was a student, I was working on a gene, and we were puzzled by having an extra RNA piece that has a size of 300 base pair, and we don't know exactly what it was, and it turned out to be actually an ALU transposon that was part of the gene, and that was astonishing, and we published a paper on it. Now, we also have another example of retrotransposons is something known as retrovirus-like elements, and they make up about 8% of the human genome. Now, so these transposons have lost in the human genome, they have lost the ability to move, to change places, but we can have some active transposable elements that can sometimes move, and when they move they can cause diseases, and there are diseases that are related to retrotransposons changing places. Examples include hemophilia A and B. Hemophilia is basically a blood disease that results in excessive bleeding, so blood does not coagulate normally, so the person keeps on bleeding. You have severe combined immunodeficiency. There is porphyria, which is related to heme metabolism. Cancers can also be caused by transposons moving. We have Duchenne muscular dystrophy, another example. So why do they cause diseases? The reason is that when you have a transposable element, when you have a transposon changing places, it can insert itself within a gene. So what it does is that it disrupts the gene. So the gene does not function anymore. Okay, and if it produces a protein, then this protein is not functional. Or it can insert itself close to a gene, so the gene is not regulated appropriately, meaning that the gene can be overexpressed, so you're producing a lot of RNA and protein, or it can be underexpressed. Okay, so there's failure to express the gene or to encode the protein. Now, otherwise, and that would cause a certain disease depending on the gene that is disrupted. Now, otherwise, the transposable element, this transposon, can insert itself far away from an important gene, and in this case, there would be no effect. But now, knowing that 75% of the human genome can be transcribed, and up to 80% of the human genome is functional, this can actually change the equation. Okay, so but that's what it is.